Our team works on the skeleton of cells, which is not a real skeleton as we know it. It is more a network of fibers which are dynamic, which grow and shrink, and which fill most of the cell, and they have a lot of very important functions in the cell. So for instance, they are important to divide a cell into two. Our team is actually investigating how these different functions of the cytoskeleton can be regulated. So we are looking into chemical processes that happen inside cells that are done by enzymes, and these enzymes can actually modify the cytoskeleton fibers, which we call microtubules, and give them a little bit different surface. And this surface is important because on these fibers there are motors, like cars, that walk along or drive along these fibers. They can transport things, they can generate forces, for instance, to make a flagellum beat. Neurons tend to degenerate if the traffic is perturbed. So we were looking into this and we found that indeed, if the modifications of the cytoskeleton we were looking at are perturbed, neurons tend to die. And as a consequence, we were thinking that there could be a link to human disease. So basically we were contacted by a group of medical doctors all over the world that had found patients in which specific enzymes involved in the cytoskeletal modifications are mutated. And together with them we could show that in these patients the modification of the cytoskeleton is perturbed. It means that the basic mechanisms we have discovered here can be valid for other neurodegenerative diseases that are much more common, like Alzheimer's disease. So what we are believing is that as we know already the enzymatic process that is perturbed in such type of neurodegeneration, we could eventually develop drugs specific to the enzymes that we know already to treat this disease in the future.